So good morning everybody. I am from Jayshree from SNS College of Technology, Department of Biomedical Engineering. So today I am going to take very interesting topic about four stages of mitosis. Okay. So we all know that DNA is under this chromosome, whereas chromosome distribute the DNA. Okay. So it is very interesting topic. Let us see in detail. Okay. So initially let us see what do you mean by chromosome. Okay. So this is what the chromosome, whereas a chromosome we distribute the DNA, right? We already seen. So here this chromosome is nothing but the centromere is present in the cell. So on this the chromosome is formed this up and down. Okay, top and bottom the two chromatid is formed, and this overall structure is what chromosome. Okay, the centromere, whereas top on chromatid formed and bottom on chromatid is formed, and this forms one chromosome. Okay, so if it is the DNA is duplicated, okay, if you want to develop the daughter cell, that it want to duplicate, right? So it need to give some cells to some chromatides to daughter cells. Okay, for this it will duplicate the cell. Okay, that is what two sister chromatides. Okay, so here it is indicates this two sister chromatides, the top two and bottom two. Okay, so here the two chromatids is formed, and the cells form this 46, where we all know that the human cells contain 46 number of chromosomes. Out of this 46, 23 are the pairs of chromosomes. Okay, this forms the basic cell structure of the chromosome. Okay, so let us see the cell division take place. Okay, whenever the cell division take place, initially the interphase is formed, that is interphase, that is cell division, that is gap 1 synthesis stage, and gap 2 is formed. Whereas in metaphase stage there are four stages are formed: prophase, we already seen that, right? Prophase, metaphase, anaphase and telophase. Okay, and finally form the cytokines. Okay, let us see the first phase of the mitosis, prophase. Okay, so this is what one nucleus, okay, one large cell, and inside this one nucleus is present. Inside this chromatin is present. Okay, this chromatin is going to form the two pair of chromosomes, which we already seen in, right? So this forms the chromosome in the stage. Okay, so this chromatin form the chromosome pair. Okay, each pair forms a chromosome. And also here the spindle fiber is going to form in this stage. Okay, it is very important first stage of the metasis. Without this, the chromatin does not condense to chromosome. Okay, and spindle fiber is formed. The spindle fiber is going to align the north pole and south pole, going to align along with this chromosome. Okay, so in metaphase is what? Next one is what? Metaphase. We already formed that. In prophase, the chromatin is formed into chromosome and spindle fiber is formed. We need to align the chromosome in the equatorial line. That is what metaphase. So this chromosome align in the single line, and this spindle fiber is formed. Okay. So the spindle fiber is formed when the chromosome is attached to the spindle fiber. It is what? Continuous spindle fiber. If the chromosome is not attached to the spindle fiber, it is what? Discontinuous spindle fiber. Okay. So in the metaphase, this formed chromosome aligned to the center of the line. It is equatorial line. Okay. That is what the stage of metaphase. So let us see the next stage is what? Anaphase. Okay. So in previous stage, the spindle fiber is aligned in the equatorial line, right? So the spindle fiber moves top and bottom. Okay. Whenever the spindle fiber moved, the chromatin also moved top and bottom. Okay. That is what this is, this stage. So top spindle fiber and bottom spindle fiber extend this chromosome to top and bottom. This forms this daughter cells. Okay. So here it is indicated by this type. The spindle fibers take the chromosome up and down. Okay. So next one is what? Telophase. So telophase is nothing but it can divide the formation of daughter cells into two. That is what telophase. Okay, the spindle fiber disintegrate, so it is a reverse process of prophase. In prophase, the chromatin is formed into chromosome and the spindle fiber is formed. Whereas in telophase, the spindle fiber disappears and the chromosome does not formed here. That's it does separate into two nucleus. Okay. The final stage is what? Cycotins. The cycotins is nothing but the two stage is divided into two. That is, plant cell undergo cycotins by forming the cell plate between the two daughter cell, and the animal cell undergo cycotins through the formation of cleavage furrow, a ring of microtubulous contact pinching the cell in half. Okay, that is what this cycotins. So this is the final stage of mitosis. So this is what the final stage of mitosis, which is indicated by this diagram. So the first, the cell division, whenever the cell division take place, first one is what? Interphase occurs, and second one is what? Mitosis occurs, and as soon as it completes G2 stage, it start the again cell to divide. Okay, so this first the cell is divided into interphase stage, and this prophase stage we already seen that chromatide is divided into chromosome, and in metaphase it is aligned into the equatorial line. In anaphase, the daughter cells are formed, and finally telophase, this daughter cells, this parent cell divided into two cells. At cycotins, finally the two daughter cells are formed. These are the all stages of that is this diagram, this graphical representation contains this all stages of mitosis. So thank you all. Have a great day.